Thank you all very much, and congratulations on finishing ahead of time, actually. And how many people thought there were fewer than 50 slides in that last presentation? More than 50? Hands. More than 75? I think there were more than 75. So thanks a lot. And do we have any questions for this session? And can you state your name, please, and where you're from?
My name is Robertson.
Is it on? Doesn't sound like it's on. Is there a button on it?
Got it now. My name is Carol Robertson Plouch, and I am just opening up a new chapter in my career, having retired early from a large pharmaceutical company. So my background is in clinical research, both in clinical research with animals and clinical research in humans over the last half of my career, with my training being in veterinary medicine. So what I want to bring up is that in this whole context of reproducibility and the context of improving our predictive value of all of our steps moving towards the human clinic, one of the things that I think that we can take advantage of or begin to recognize more fully is the opportunity to leverage the diseases that occur in our companion animals, our pets, such as dogs and cats, that are very similar to some of the diseases that we get. And that is a very underutilized opportunity, but something that I, in my last foray in the pharmaceutical world, had initiated at that, with that company. And in that way, by combining the data that we have existing and available through our conventional laboratory models, and then also, when it's appropriate, introducing a real-life disease from an animal that may have a disease that's more fully recapitulating some of our diseases, it should help us to be more predictive of what's going to work in humans. And there's been a few examples where this has been done quite successfully, but I think we need to flesh this out even further, and I'm hoping that in future discussions that this concept can be introduced in this forum.
Would any of the panelists like to respond to that?
Yeah, thank you for the comment. I couldn't agree more. And I know in the Netherlands, Utrecht University has a program which is called One Medicine or One Health. And that is exactly aimed for using veterinary patients for the purpose you mentioned. So thank you.
And there are initiatives here as well, and I think we have some speakers that represent some of them. And
hi there, I'm Sarah Wells from the MRC Harlan in the UK. I was slightly disappointed to see on a slide saying that there's been no improvement in reporting or adherence to the arrived guidelines in 2017. Running a large facility, I can certainly say that there's quite a disconnect here, because I think more than ever there's a big concentration on experimental design and on reporting. And I think maybe we're kind of getting towards a situation where on the ground it's very much improving. There are training courses for PhD students, like many more than ever before. I think there's a much higher focus on experimental design. Definitely, I'm proud to be part of a large consortium who adhere the IMPC who adhere very strictly to the arrived guidelines. But I would say that there is a little bit of a disconnect here as to what role the journals, peer review and funders have into ensuring that the reporting is done properly, and that we don't publish studies that don't adhere by those guidelines or good experimental design.
do any of the panelists want to respond to that, or all of them?
Well, let me just ask a question. How many people here have heard of Arrive? Wow, that's impressive, because most of the time when I speak to scientists, there's like 3 hands that go up, and it's usually the person that invited me. But anyway.
No, I don't think that's true.
I'm telling you what my own experience is, and
It's taught
From the U.S. perspective, I see far less awareness of it here in the U.S. than I do overseas. So far,
I might just
optimistic.
add that I just recently stepped down after 9 years as an editor of a journal in the field of stroke and cerebrovascular medicine, and I think we were among the first to adopt the ARRIVE guidelines of all journals, and we started by having a check sheet that's where authors had to check comply with ARRIVE. The day we instituted this policy, we got a 100% list of ticks. People didn't even know at that point what ARRIVE was, but they were fully in compliance. Then we got smarter, and we started to have a 4-page questionnaire that had most of the ARRIVE points, and people they all complied and on those check sheets. And I was still confused. And then I started to pay students to go through all the submissions and go through the papers and, to the articles and check where you find the because part of ARRIVE is that it tells you to write it in the paper, not on a check sheet. And it turned out that in more than 70%, and this is still 2 years ago, more than 70% did on the check sheet comply, but not in the if you really checked. But I take it. I think it's a good. There are good news. It is improving, and so. But you may have seen Emily Senna's. Was it pre-register? Is it a full publication now, or is it an?
No, it's
PowerPoint. Yeah, and I think it's in bioRxiv or somewhere. And that was actually a randomized trial that they did on this, and so the numbers do not really reflect what you just said, that the uptake is much better. So that's I wish you were right, but the numbers speak different from a different reality. We still need to work on it. I think it's getting better.
I think Dr. Valley was next.
Can I make one comment? Because we also at the round table last week, we had David Mower on the Skype from Canada, who knows a lot about implementation science. And he said for journals, it is really important to write clearly what they want. So by using words like endorse ARRIVE, that's too vague. So the journals should really state what it means when they want to take ARRIVE, and that authors follow the ARRIVE guidelines.
I wanted to ask what I think are two related questions. And forgive me, I'm not a biostatistician, so I may have gotten lost in the weeds somewhere. But much of the last two talks described scenarios where the patient population was fairly large, and a standard approach would be to go to a randomized clinical trial to try to help evaluate the reliability of a prediction. In many instances, in genetics, we start with patient groups that are actually quite small, quite rare. And the design of randomized controlled trials is pretty difficult. And then, as I thought about the talks that we just heard, in my view at least, the pursuit of individualized medicine, we will always be going to smaller and smaller groups of patients because we're seeking individuals that are more homogeneous and more like one another than so that it seems to me we'll be losing power as we go forward, even amongst the phenotypes that at the outset seem pretty common. And we'll be gaining homogeneity, but we'll be losing power. So how are we going to handle that? And if you said that and I missed it, please forgive me, but I'd be interested in your thoughts.
I thought that was exactly what I said. No, it's great. It's great. You summarized it better than you said it in one sentence better than I did in 20 minutes. And what I tried to suggest is that, I don't have a simple answer. I think that probably if we are losing power, I'm not a statistician, by the way, so I'm going to defer, to Arjun, but let me just muddle my way through this. It seems to me that we are used to in medicine, maybe not in rare disease, trying to combine evidence streams where the evidence is really, really similar. The evidence is all collected from randomized controlled trials, the meta-analysis. It seems like the task when you're dealing with small populations is to combine different types of evidence. They can't necessarily be pooled in the way that we do a traditional meta-analysis. If you think of what a weather forecaster is doing, weather forecasters don't do a meta-analysis when they make a weather forecast. They're looking at a bunch of different kinds of information. They're looking at ground conditions, climate conditions, right now. They're looking at various models that are simulating what's going to happen. And they're looking at maybe historical, what has historically happened, under these various conditions. And they're combining these different evidence streams into some kind of a judgment about what's going to happen. And what I was trying to suggest as sort of my third recommendation is that it may be that we need to think about training people to be more effective consumers and aggregators of information and evidence.
So I would agree with that, and I think what you're describing is probably the fundamental tension in precision medicine, which is that you want a very well characterized, very well defined population, but then you're left with 5 people. How can you make a robust claim from observing a particular mutation or seeing the response to a drug in a case report? I think there are two responses. So broadly, I think this aggregating or binning or sort of collecting different types of evidence, I think is cause for optimism. So I think while it might not be possible to rigorously evaluate the causality of a given variant in a particular family, the strategy itself across the patient pool might be able to be evaluated. I think the Shiva trial is a good step, but I think there's only one of those, as Professor Kimmelman pointed out. I think the second is that evidence comes from other sources in very surprising ways. And so in the context of human genetics, this is, for example, the disease that we study a lot is hypertrophic cardiomyopathy, 1 in 500 prevalence, inherited thickening of the left ventricle of the heart. Genetic testing has been in place for at least 10 years for that disease, and every family has a different mutation. And the paradigm, both from how statistical it is, how rigorous it is completely different than the GWAS world and the statistical genetics world. And I think where those two have merged over the past couple of years, and we see this now all over the place, is with the proliferation of these very large databases. And so NOMAD, EXAC, these are, both of those are from the Broad Institute. I think NOMAD is a genome aggregation database. It's 140,000 individuals. In the New England Journal paper that we published last year, we used a data set from the NHLBI called Exome Sequence Project, which was 6,500 individuals. And these data sets, I think, would have been, you wouldn't have been able to imagine that they allow you to reevaluate the candidacy or the pathogenicity of mutations that were considered causal for several years, just a couple of years ago. And so I think that is cause for optimism, but I think the statistical challenges of making robust claims about very small groups, no one has a very good solution for that. It's by construction, I think, a very, very hard problem to solve well.
So it suggests to me that, as is the case, I guess, for any good scientist, that we could make predictions and we could realize the level of strength of our predictions, and then we should do some sort of ideally orthogonal test of the prediction to sort of gain additional confidence based on our prediction. After all, the physician will be sitting in the room with a single patient and helping that patient or making for that patient some decision.
I wanted to make one short comment. I'm by no means a statistician and not involved in the clinical scene, but I have colleagues who are working on individual patient data meta-analysis, and I think that is particularly meant for having data of patients to see what is individually predictive. And so I think the Cochrane also works on that. So I can bring you into touch with people if you want.
But I think your point is very well taken, which is that there's information that's being thrown away that is not coming back into the sort of the calibration of the model itself, right? So I think genetic testing is what I'm very familiar with. When we make a prediction in the family, typically the geneticist will sequence the proband, and if they have a familial mutation, they'll use it to test all first-degree relatives. So, for example, 2 sons of a patient with hypertrophic cardiomyopathy. There's the follow-up, whether they actually go on to express the disease, because at that point it's essentially just saying that you're at risk of it, and if you don't have the mutation, you're not at risk of it. So there's no clinical sensitivity or specificity, and I think the actual calibration of the model itself, which is does this given individual go on to express the disease itself, that information, there's not really a good system still of integrating that. I think steps we've taken, there's a couple of labs that have very detailed records, so we can do retrospective analyses. Now it's at least 10 years old, so we have some type of data to make claims like that, but I think completing that feedback loop, with the weather, you can look outside and you can test your model that way. But here, I think we need ways of getting that information longitudinally back into the original, so we can know what worked and what didn't work. So.
Dr. Peterson?
Norman Peterson, MedImmune. So drawing on the previous session and perhaps bioinformatics from this session, I think the way that we cure disease is either at the protein or molecular level. But when we were talking a lot about the phenotype, we phenotype our models at the morphological or physiological level. And I'm just wondering if it's time to kind of rethink the way that we define a model and come more down from this mile-high morphology down to the level of where we work and look more at the microenvironment, both in the human patient and in our animal models. I'm just wondering how practical it would be to define the microenvironment at sort of using either proteomics or genomics to define the characteristics or phenotype the models at that level, because that's the level that we're working to target them, and then use the bioinformatics technology and databases to make sure that those correlate and relate that back to our treatments.
I think you're right. It needs a lot of informatics, because we get so much information out of the omics scene that I think nobody can combine those things except for a computer with sophisticated software.
How about a weatherman?
He probably has computer programs as well, doesn't he? Yeah.
Jim Stevens, Eli Lilly. I hope this isn't a hopeless naive question, but I want to address this concept of reproducibility.
I think you may have lost your... Okay, there you go.
Jim Stevens, Eli Lilly. I want to address this concept of lack of reproducibility. You can think of 2 kinds of situations where you can't reproduce something. One is it's a highly random system, and if your likelihood of having true positives within the data set is very, very low, then you'll always generate a lot of false positives because your specificity will never be high enough. In that case, you'd think the same person doing the same experiment would get a different result every time, because it'd be random. So the question I want to ask is, when you look at lack of reproducibility, do you find that there's high reproducibility within a lab, but lack of reproducibility across labs, or do you find there's a lack of reproducibility within the lab? And let's exclude, poor experimental practice, unethical behavior, et cetera. Because if it's the difference between, is a lack of reproducibility due to a sensitive system where environmental factors play a major role in the lack of reproducibility, therefore it'd be highly reproducible within the lab, or is it truly randomness within the system? The same lab doing the experiment over and over just gets a different result every time. Does that make sense?
Yeah, I think that's...
How do you tell the difference?
Yeah, I think it's probably impossible to tell the difference, and I think it has a lot to do with the base rate of our investigations. I mean, the prior probability whether our hypotheses are right or wrong, and this is what we don't know that. And the base rate, I guess, of most of the experimental science that we do is very low. It may be in the percentage range, and if this is the case, your scenario 1 is what we are looking at, and this would be testable in the sense that in your example, the within laboratory reproducibility would be low, should be low. The problem is we don't have data on this because no one reproduces own data.
But assuming reasonable levels of laboratory practice and adherence to standards, my guess would be that most laboratories do in effect reproduce their own results. Yet when you go across labs, you find this lack of reproducibility, and it seems to me that tells you that the experimental system is highly context dependent.
Sure, yes.
But it's not a lack of reproducibility, it's an oversensitivity to environmental factors. So somebody uses the cell line at passage number 210, somebody uses it past number 150, and when you get from 150 to 210, you have a completely different cell line. You're doing the same experiment, you get a different result.
But it also has to do with how standardized the environmental factors are. If you think about buying commercial chows for doing animal studies, they will be different from batch to batch. So if a result is very dependent on the components in the diet, you will also get no reproducibility within one lab. Whereas if you would use purified diets, then you have a high chance of getting a reproducible result both in your own lab as in other labs.
Yeah, but a probably quite useless result because it's dependent on one chow manufacturer.
Dr. Brown wants to make a comment about the IMPC experience.
So I sense this conversation is on a slippery slope to a lot of negativity about how we can, the ultimate goal is to define mechanisms, underlying bases, which, to go back to, I think somebody mentioned Karl Popper, withstand constant testing, that we can be sure that this is the way the world works. And so reproducibility, not only within labs, but across labs, is fundamentally important, and if we can't do that, I would suggest we're pretty lost. But the IMPC have done quite a lot of this work. I didn't have time to mention it in my talk, but we take particular mutant strains and we put them through the pipeline at the different centers across the world. We put a lot of emphasis on first of all, standardizing the operating procedures that we use, which is fundamental, and in particular recording all of the metadata down to the person who carried out the phenotyping, down to the feed, down to the environmental conditions, and so on. Through those processes, we find a very high degree of concordance in the data that we get from different centers. It is reproducible, not only within centers, but also across centers as well. So I think we can be more positive, but it's very hard work. It's very hard work, and we know from programs that preceded IMPC, there's a big European consortium that worked on this, worked for many years on the operating procedures, testing different inbred strains as controls to make sure that we got the same data in the same centers, both across place and across time. So I think we can do it, but it requires a sense of rigor and determination and just bloody hard work to get that rigor. But I think unless we strive for that, and reproducibility is important, then everything that we're trying to do in terms of the precision medicine initiative will be lost. So let's put it this way, I'm more optimistic that we can get over what we might think of as a rather sensitive system that's difficult to reproduce across laboratories and find solid, reproducible data about how cells, organisms work.
Thank you.
There's a factor that I don't think has been discussed much.
Is this Dr. Chow?
Keith Chang, Penn State. So one of the factors that I see active is there's a lot of pressure, of course, to publish for PI, for grants, for students to get the same finding that the rest of the group wants. And if you could make the reward system include reproducibility factors, and in publications or otherwise, such as you guys have developed some of the tools that we might be able to use. I think that might help things. If students know going in, faculty know going in, that it matters that the work is reproducible, and they might produce orthogonal data for their findings, then I think they're more likely to succeed.
I just wanted to say a couple things. Back to the earlier comment about can we just create precision models based on omics profiling of an individual cell type, or genomics or metabolomics, or whatever your favorite flavor of omics is. And I think this speaks to both one of the challenges for the informaticians, but also for reproducibility issues, is that fundamentally we're doing a very poor job of relating those omics data to the phenotypic manifestations that happen at the cellular, tissue, organ, and organismal level. Those outcomes are the disease states that we're observing in the patients in the clinic and that we're measuring, but we're not relating those very well at a fundamental level, sort of mechanistically in our models. And I think we have to kind of address some of those informatics barriers before we're going to be able to best do that kind of precision modeling, as well as to kind of use that same kind of infrastructure for the reproducibility aspects. My second comment, which is a really simple one, is that this has been about 4 years ago. We actually published a paper where, as a research data parasite, pretty classic. I fundamentally rely on the literature and all the public databases that publish data that go through the literature and look for, how to curate the information. How do we actually capture the information? And that's a reproducibility issue as well. If we can't get it out of the literature and into places where it's computable, we can't actually use it. 50% of the model organisms that we reviewed in this manuscript were not uniquely identified. I couldn't figure out what data to attach them to. So, reproducibility, is all kinds of different things. But if I can't even figure out what the reagents were from the experiment, then it's a non-starter. So those are two kind of unrelated points.
Okay. So we're about 10 minutes late, but I think that's a good thing. So that we had some questions and we had some discussion, and I hope you'll think up more questions for tomorrow, and you'll have an opportunity to speak with most of the speakers tomorrow as well. So thanks very much for participating. And